Zašto? Evo ga ovdje, znači ovo nam je to dugme. I sad kad imam dijeli ekran, ovdje entire screen, boom, trebalo bi ide. Sad idemo... Čekaj sad, kako idem sada... Composition, yes. I sad sam... Evo,
Rado.
Oh, yes, it's nice. I'm sure that's my mom and my favorite. That's good. I think I owe you one. Thank you. Is there already so much in here?
Yes, I think.
Looking for clerks in case.
Will be the last session, so.
Mm-hmm.
Okay. Over there, I guess.
Okay.
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 Thank you.
Because my presentation when I was here, they want to use this one. Yes. Yes, yes, it's okay. No, I don't know if
How are you? Okay. Yeah, it's actually more or less the same. Okay, because that one has got a problem. I guess it probably is. I know. Now you have to carry the two bags for the rest of the day, yeah.
Because she said the first mistake she said, and I thought she said, well, they're fine. So, from UK, I guess. I don't know exactly what. I think.
So how about this Epicatch things?
You make so much like a Epicatch,
The letters, yeah. I know, but still, yeah. But still, it's good. You have to be. Well, it was national TV. Well, we said something. It's about getting so inside the program and everything, changes the book of everything inside. Yeah, I mean, I'm satisfied. Yeah, each one of the conference also. Yeah. What do we give her that? Don't expect too much. I have to tell you, don't expect too much. Be gentle with us. Yeah? Okay. So you have like Wi-Fi over there, yeah? Schools and everything. So here is the program and here is the Wi-Fi. Irina sad čeka u šareni haljini u ognja. A verovatno.
Try to see if it's good. C'est bien, c'est beaucoup, c'est de
Yes. No, it's okay.
No, I didn't buy anything, so I checked it.
How are you? See you. Yeah, good. Okay. I came yesterday.
Yes, you succeed to find see a book or something?
Yeah, I organized this.
Okay. Bonjour. Bonjour. Ça va? Because I don't see your name. There was one of us.
The second speaker.
Oh, yes. It was for my passport. Yes. Because of the production. It was a small bus and about 20.
So I organized this with I was going to use it. I organized with that company that was with one staff.
I can go at the same time as your right lesson. So half past something like that. Yes, because someone told me that if he saw you, you see that.
Irene dava presentazione.
Okay, that's perfect. The only thing is to find the way to complete it.
Yes.
Should I open it to check if it's real?
If you want.
Yeah. Yeah.
Wow. Okay, okay. This is.
I know. Just until the people come, okay? Then I will tell you so. No, it's okay.
Is the presentation today or?
Yeah, it's for today. I don't know what time exactly. But this afternoon for sure. So I think it's called Epicatch something. Yeah, Verdi. And can I check with you because sometimes I have a figure that doesn't appear well. I mean now it's too late anyway, but
You never know.
I don't know why sometimes. I think this one or this one. It works perfect. Very good, thanks.
Oh, wait a minute, just so I can return you your USB.
Oh my God. Did you register? I told you to come at one. It's usually come.
She's not there.
Oh,
Okay, sorry.
no, it's okay.
We can check. I have some movies, but if they don't work, it's okay. It's not.
Okay. Your presentation is today.
Yes.
This one?
Nice electric theater.
Thank you. Is that it? Okay.
Oh, thank you. Is it okay to open it to
Yes.
double check? Because it's in a Mac, but I must have pressed something that I shouldn't have. I just want to see if, it may not be working, but it doesn't matter. It's okay. There's a movie here, but okay. Anyway, it's fine. Great. So just close it. Great. Thank you so much.
You're welcome.
Yeah, I think for me that's the worst part. No. It's okay for that. So I think everything's as one because at least we have so there's a period of another. So probably but the quality is really bad. So the applicants, they are back in Vietnam, it's already so it's all depends on the applicant. So only, you can get the money. Because they have this deal and they want to, I think they sign a contract saying you buy the heaven, but you can't buy anywhere. Then you cannot do any changes anymore. Hey! Is your presentation like this?
Yeah. You want to put video? So you. Every bucket is water. Yeah. Did you know? Yeah. They have prepared everything for us. So he was going here with me. So you collaborate with me? Yeah, it's this new development. I mean, you're using traffic. I'm working with kids. 15 minutes I cut it. Oh, that's that's 15 minutes in total, yeah? And then we will start the 45. Okay? So we will for next time I will leave, yeah? So you have to say something. Hi, Marcus. Thank you very much. Yeah. So, I'm from Germany, yeah, but he's from Germany. He's working in. He's working in. Do I look like German? Yeah, a little bit, yes, but I don't think so I work, so do you know people from the I'm not stress-free. He's the connection with his I'm not stressed at all. I will say no. Me, I'm just he's a new generation. The new kid in his life. Yes. Soon. No. But I was working with the last because I don't know if you know. Yeah. He owns the first registration Last because and each note. I met the HSF factor from University of dinner with him. This? Yeah. The beer. Yeah.
Time من أنا
Yeah. Indeed. It has been nice, but it was not. It's working. Yeah, it is. And he's been working. I didn't know. Yeah. How do I do this, Aki?
I don't know.
You don't know, huh?
What do you want to do?
I
Yeah,
don't know.
here.
To share this one.
Yep. And full screen.
Where? Here?
Full screen here.
All right.
Where do we sit there?
Yeah, there at the beginning there.
So on behalf of the Epicatch construction, I welcome all of you in Novi Sad. Before we start, I want to thank the local organizers, and especially Professor Pedro for all the fantastic work that they have done. We are in a beautiful room here, and they have prepared for us a fantastic program, very interesting 3 days of science and networking. This is the maybe the last year of Epicatch, but we hope that this is something will come out and more Epicatches come in the future. And I would like to welcome the chair, the most important person in the room, Federico Martin. You are. I'm Greek, you're Italian. Federico Martinelli from the University of Florence, and he will give an introduction, I think, about Epicatch.
Yes.
What it is about, what we've done, and get inspiration for a new Epicatch in the future.
Yes, yes. So thanks again to all the organizers, but especially Vladimir that organized this and give us the opportunity to visit this beautiful city. Really, thanks. I'm sure that we will have 3 days of very great enjoyment between science and other things. So just 5 minutes to show you the things that we have done in Epicatch in this year, basically. Here we have done several events in 2024. As you know, we are a cost action that deals with the epigenetic mechanism of crop adaptation to climate change. And as Aki's already said, we are at the end of the cost action, unfortunately. We have only 2 months left. What is a cost action? For those people that doesn't know, cost action is a program funded by European Union that funds ideas and connect people. Basically, the project is 4 years. Every year they give you a budget for organizing networking activities like short-term scientific mission, training, conference, workshops. With the aim of establish long-term collaboration on new ideas, product through these activities. Here is just a map of the different countries involved in cost action. As you see, there are 2 kind of groups, the cost members, and then there are the IT's countries. So when you develop a project, you need to be very careful in following the rules for involvement of different countries. What we address in our cost action, we basically address 3 challenges in plant epigenetics. One challenge was, as you guys know, the need of investigating epigenetic responses of plants to environmental stress, and then also define new frontiers, new innovative aspects in plant epigenetics. And the third challenge we identified was trying to improve lab and in silico data analysis in plant epigenetics. So basically, we structure our cost action in 4 working groups. Every working group tried to address these 3 challenges. And then there was also the working group 4, basically the core group of the cost action that helps the other people of the other working group to organize the events on these different aspects. Just very few details about the key aspects that we covered. In the working group 1, as I told, we are dealing with plant epigenetic response to environmental stress. So we cover drought, salinity, of course, heat temperature, biotic stress, and also heavy metals. In the second working group is dealing with new concepts and new frontiers. So we organized events on transgenerational stress memories, but also we try to identify common epigenetic marks among different environmental stress. And then we gain insight in the complex epigenetic code alphabet of plant stress response. Here we, with the help of some reviews that we wrote. In the third one, the challenge on methods, we wrote a review on best practice in plant epigenetics and also on the methodological aspects. So just a very few words on the events that are ongoing, future, and our past events in 2024. We organized a training school in different countries. It was an international training school. It was organized by Livia Stavolone and Andreas Giselle. It was really a great event. Was a hybrid event in different locations. It was dealing with in silico analysis of plant epigenetic data, and then a workshop was organized by Ariola Baku, that she organized a workshop on different aspects in plant epigenetics. Then this current event organized by all your organizing committee and Velimir, and then we are planning 2 other events. So for those people, if they are still interested to participate, there is an event in Bordeaux. It will be 10-12 July. Will be organized by Philippe Galusci and Nadia Pons. There are, I think they accept still registration. So if you are interested, just go on the website to register. And I am organizing a workshop in Florence, the 2021 June of 2024. And still, I need to fill the program. So please contact me. Please contact me if you still are interested to make some oral talks. Just contact me. We have only very few time left, but still I have space to have some of you. Here is our website for any update between until the end of this cost action. And if you are still interested, there are some months left, but if you want to be officially a member of our cost action, just go on the link, follow the instruction, and contact me for any questions. Here again, thanks again for Velimir again for giving us the chance to spend these beautiful 3 days in Novi Sad.
Thank you, Federico, for the honor that we can organize the conference here. And now the Professor and Vice Dean for Education, Mirjana Đukić, will open the conference.
Thank you. Dear guests, dear colleagues, welcome to the University of Novi Sad, Faculty of Agriculture. It's a great pleasure, my pleasure and honor to have you here, and this also epigenetic cost conference. Cost actions are always good opportunities for networking, for establishing new cooperation and partnerships. I wish you a successful meeting those days and pleasant stay in Novi Sad. Velimir, my colleagues and I, we are here at your disposal, and do not hesitate, please, to ask anything. Thank you.
Okay, now I'm very pleased to open the first session with the most important stress of all, heat stress, right? And if you don't believe it, we can turn off the air conditioning. So I would like to introduce Professor Musa Mohamed from the University in Paris. He's
a pointer
an
or
expert,
not? Because if you do,
from
then I have
he
my.
will talk about changes.
And do you have a pointer? To show the, can I use
organization
mine?
use you under high temperatures.
Sempre se qual é a apresentação?
Fora lá, se me...
Qual?
Fora tudo. Voilà.
This one, Musa?
Yeah. And if I can plug this, can I put it here?
Yeah.
Yeah.
Hello, everyone. It's a great pleasure to be here in the front of you. And of course, I would like to thank the organizer for giving me the opportunity to present you the work that we have done recently in the lab about the interaction of regulatory elements, mainly enhancer with promoter, and how those interactions between enhancer and promoter can impact gene expression in tomato. And the work that we have done, as Aki said, it was done in the context of heat stress. But before we start and going in deep, of course, a little bit of background. Whatever the eukaryote, can be human, mouse, Drosophila, plant, all the eukaryotes, when they face an environmental or a developmental stimulus, they all do the same things. Like part of the response, the physiological response, will be driven by this transcriptome reprogramming. And one of the main questions we ask in my lab is how this transcriptome reprogramming could occur. What are the mechanisms behind this big transcriptome reprogramming? And what we know, as I told you, it can be whatever the eukaryote. When they perceive a signal, then of course you have all of the signaling cascade that at the end will be integrated inside of the nucleus. And this will induce changes in the 3D structure of the chromatin. And this 3D structure that will change will allow the regulation of gene expression, and will allow the right gene to be expressed at the right time, if you want to say it simply. And in the lab, what we are interested on, of course, is about this chromatin structure. And usually when people talk about chromatin, they show it this way. Like you have what we call euchromatin, which is a relaxed form of chromatin. You have here the histones in blue, the DNA in red. And these relaxed forms of chromatin allows the binding of different transcription factors. And you can have also this heterochromatin, which is a much more compact form of chromatin that doesn't allow chromatin to be accessible for a transcription factor. And you have different mechanisms that will allow the switch from one form to another, like the histone acetylation, histone methylation, the action of chromatin remodeling complexes that will use the energy of ATP to change this structure, and also DNA methylation. All of those things will be there to, let's say, dynamically change the chromatin structure and to control the DNA accessibility. But when you show this, you show basically what happens at the nucleosomal scale. And if you look to only what's happened at the nucleosomal scale, it's very difficult to understand this transcriptome reprogramming. In fact, if you look only to the nucleosomal scale and you see just if chromatin is more or less compact, you can understand how one gene is expressed, but it's very difficult to understand how many genes could be co-expressed or co-induced by a signal or co-repressed also. In fact, what we know is that you have to take into account not only what's happened at the nucleosomal scale, but you have also to take into account the interaction between enhancers and promoters, or repressors and promoters also. You have to take into account that inside of the nucleus, chromatin is highly organized. You have regions where genes are, let's say, transcribed, but you have also regions inside of the nucleus where you have no transcription because you have no transcription factor available there. You have kind of microenvironment there. And we have to understand, if we want to fully understand this transcriptome reprogramming, we have to check what's happened at the nuclear scale and how dynamically this chromatin is reorganized. Because those days, it is clear, you cannot read because of this, but the chromatin topology, like the 3D organization, is key for the reprogramming in response to stress. Some few details, because when I show you this picture, this is for whatever you carry. Now we will go a little bit more in details. If you look at this level that we call the intra and interchromatin domain contact, you have different, in fact, inside of the nucleus, to say it very, let's say, easy, you have two types of compartments. You have compartment where you have the genes that are transcriptionally active, and you have a compartment where you have the genes that are silent. And this compartmentalization is made thanks to different proteins in mammals, for example, thanks to coenzyme, thanks to CTCF, but in plants we don't have CTCF. And for the coenzyme, no one knows really what could be the function in plant. There will be some papers that will come soon, but it's not something very clear. And in animal, you have also some, let's say, structure that we call topologically associative domain that are, let's say, genes that are grouped together to be co-regulated. And this was defined first in animal. We saw something that looks to be similar in plants, but since they are not exactly the same, we call it that-like. In any case, just to give you this overview, for one reason, to show you, to try to convince you that this 3D organization is key. And in my lab, the major question we ask is how does chromatin dynamics orchestrate plant development and response to stress? And how do we study this? We study this basically by generating transcriptomic data, epigenomic data, and also analyzing the 3D structure of the nucleus using molecular biology and integrate all of this. Basically, we try to integrate as much as we can epigenomics, genomics data to decipher mechanisms. And we do this in the context of abiotic stress, namely heat stress, with Akis, in collaboration with Akis. We do also in the context of biotic stress, plant development, and we study this also in wheat. Today I will talk about the work we have done in abiotic stress, and this was done by a PhD student in my lab in Bonn. What we use as a model system for this abiotic is tomato. Of course, all of you know why tomato is important. Tomato is important for agriculture. But the first question in the lab, when we started working on tomato, was just a simple question. How is tomato genome organized in 3D? Because there was no publication about this. Then what we did what scientists like to do. Do experiments that you can see results with your eyes. Not do molecular biology where you can put a drop of this or drop of that in a tube and you don't see anything. And you have to trust. We wanted to see. Then you did immunostaining. We use antibody that requires specific histone modification. Here, for example, is an antibody that recognizes H3K9 Me2, which is the histone H3 demethylated on the lysine 9. This is a well-known mark of histone associated with constitutive heterochromatin, which is mainly TE. And this is a width nucleus here with the dye. This is the same width nucleus with this signal. What you see, everything is associated with the nuclear envelope, okay? Which means that the TEs and the chromatin that is silenced, that we call constitutive heterochromatin, is massively associated with the nuclear envelope. We use an antibody against H3K9AC. This lysine 9, when it is acetylated, is associated with gene expression. Very well known. And here you see dots inside of the nucleus, which means this is where you have genes that are expressed. Okay? You see this compartmentalization very well when you overlap this. And we use several other histone marks, and we did a lot of immunostaining. This I showed you just one, which is H3K27Me3. This histone mark is associated with developmental genes that are repressed. And you see the developmental genes that are repressed are inside also the nucleus. But not associated, of course, those genes are not in the same region where the one are expressed and not associated with the T. Basically, you have a high organization. You have a high structural organization inside of the nucleus. Again, the constitutive heterochromatin in the periphery, genes that are transcribed in the center, and genes that are repressed also in the center, forming kind of clusters. Okay, the second question we ask after that, how this chromatin structure could be affected by heat stress. And of course, we didn't want to spend a lot of money for nothing. We wanted to do a simple experiment that can show us that, yes, we can have a chance to see some changes. And then what we just measure the nuclear size before and after heat stress. We did the heat stress for 1 hour, 6 hours in tomato. We measured the tomato nuclear size, and what we saw, after 1 hour of nucleus, the nuclear size increased. After 6 hours, you start seeing a decrease compared to control. You start seeing at 6 hours compared to 1 hour, but still a little bit higher than the control condition. What does it mean? It means that you have changes that happen inside of the nucleus. The question is, what are those changes? What could be the impact of heat stress on tomato chromatin architecture? And the question is how to see it. But this time we need molecular biology technique. And what we use a technique that we call a high-C. What is a high-C? High-C is a method that allows you to know genome-wide which piece of DNA is interacting with which piece of DNA inside of the nucleus. How it works. You isolate nuclei, you cross-link everything with formaldehyde, then everything is fixed. You do this in controlled condition after 1 hour of heat stress or 6 hours of heat stress. You do your cross-linking. Then after your cross-linking, you digest everything. After your digestion with restriction enzyme, you fill the end, and repair this and add a specific biotinylated nucleotide. You make ligation. Everything that is close to each other will be related. And then you create chimeric piece of DNA. And what you do, you will sequence those chimeric piece of DNA. And what will be the output of this after parent sequencing? It's a contact matrix. How you read this? Here you have the genomic position of one chromosome. You have the same genomic position of the same chromosome. When you sequence your reads, you sequence them parent. Then you get the sequence from one end and sequence from the other end. And what you do, each sequence, you map it on your matrix. Like this end goes to this matrix if it is at this position. The second end, you see it from this axis at this position, then you get one reads here. If chromatin inside of the nucleus, we do this for all the things, if chromatin is linear, you get this signal. Okay? And this is what if you get this signal, this is how you can see your chromatin structure. But in fact, when you do this experiment with tomato, and when you do it with whatever eukaryote, you see the same things. You see, of course, this strong diagonal, but you see also something that is very strong outside of the diagonal, which means that the chromatin is organized, of course, in 3D. And then the question is, what will be the impact of heat stress? We use this technique, of course, in the lab. And when you use this technique, you can compare. Here, you have a heat map of the high C. Half is in controlled condition. The other half is 1 hour after heat stress. When it's red, means that you interact. When it's black, means that there is no interaction. If you zoom in this region, which is a region where the density of gene is very poor, what you see red things, for example, here. In the opposite side, this is in control condition. Here, after 1 hour of heat stress, it's black, which means that if it's red here, it means that this region is interacting with this region. Whatever, you have long-range interaction. Here, you don't have it anymore. And you can quantify contact frequency among the genomic distance. This line in gray is the control. The blue and the red is after 1 hour or 6 hours of heat stress. Another way, how you can see this. This is your chromosome in tomato. You have your pericentrometric region. You have regions that are very high, that are very red. Here is where you have the genes. Here, gene poor region. In controlled condition, everything is very well packed, to say it simply. After heat stress, it starts to relax. And this could explain why the size of the nucleus is higher, because you have a kind of a relaxation of the chromatin structure inside. But what I show you here that happens in tomato, and not unfortunately, I have to say it was already known in Arabidopsis, nothing new when we did it. Then we say, okay, Heat stress causes decrease of chromatin interaction in constitutive heterochromatin, but nothing new. Then we started, okay, we should look to region where you have the genes. Where you have the genes that are up and down, what will happen there? And when we analyzed this at the resolution we had, we saw almost nothing. And we started to be a little bit disappointed. Here is the contact frequency with a function of the distance. In controlled condition, after 1 hour of heat stress or 6 hours of heat stress, you don't see much difference. A little bit difference here, like the chromatin interaction on the euchromatin were slightly enhanced, but not too much. And me, my student, mainly my student, to say the truth, me a little bit, but my students were really upset and unhappy. And she said, okay, I spent a lot of time, but it looks like we see nothing. And then I told her, well, look, we focus on the region where we know that there are genes that are up and down and see what we can get with these data. Then we combine our high C with the RNA-seq. This is another way to see the high C data. On the top here, you have the genes. Here is the RNA-seq. Those genes are not expressed in controlled condition. This is the result of the high C. More the color is intense, more you have interactions. Here you have RNA-seq after 1 hour of heat stress. For example, this gene highly upregulated. This one also. But when you compare these and these, to say the truth, no massive changes. And if you look to 6 hours, those genes get down and we don't see much difference. And we have two explanations. Either there is no changes, or it was the, let's say, technique we used that didn't allow it to get a resolution that could be enough to allow us to see major changes. Why? Because here the resolution is 5 kb. And it could be that what happens is changing in terms of interaction between genes and regulatory elements that are inside of those 5 kb. And we don't see with this data. Then, you have two options. Either you leave this in a corner in your computer, or you start to continue to move on. Then we say, okay, we will be positive. We will still think that there is changes. And we thought that the changes could be inside between regulatory elements and genes. Regulatory elements, you have different types of regulatory. We define them as proximal, if they are very close to the transcription start site, and for us, very close means 1 kb. If you are more far than 1 kb, we call it distal regulatory elements. The first question, there is no atlas of regulatory elements. Then how to fish them? Then we use a technique that we call ATAC-seq. ATAC-seq is a technique that allows us to fish all the regulatory elements in the genome. How? These techniques relies on the fact that you use a transposome. This transposome can integrate in the genome where chromatin is accessible. And chromatin, the piece of DNA that are accessible, are the ones that are bound by transcription factor. Because transcription factor, most of them, they cannot bound chromatin if they are associated with nucleosome. Then we use this ATAC-seq, and we did it in control condition after 1 hour and 6 hours. Then what we found? We got this kind of map. When you look like this, you say, oh, this map is really strange and not easy to read, but you will see it's very easy. This map shows you what? In blue, each square in the map is a regulatory element. When the regulatory element is blue, it means that it's completely closed. When it is red, it means that it's open and active. What you see here, basically, you have several regulatory elements that are not active in control condition, very active after 1 hour of heat stress, and then start little by little to be less active. And when you see this, you can generate cluster. Control condition, all the regulatory elements that are open, but then close after 1 or 6 hours. You have regulatory elements that are closed only at 1 hour after heat stress. You have other, and this is for us the most important, you have regulatory elements that become accessible only after 1 hour of heat stress. And with this regulatory element, when you find them, you look to the sequence of DNA. When you look to the sequence of DNA, you can find the motif associated to them. And then the family of transcription factors that are active. Here, for example, in this cluster, what kind of family of transcription factors are active? The HSF. This is what you expect. HSF are well known to be active in the primary response to heat stress. Then you have a very dynamic chromatin accessibility that occurs, which means that if you have a lot of chromatin of regulatory elements that become accessible, they have to be able to be close in proximity with the genes that they can target or they can control. Then, when we saw this, we said, okay, we have two questions. You have a regulatory element, like this one, that can be far from this gene and far from this gene or at equal distance. The first question is how to connect those regulatory elements to the right gene. And the second, we have two types of regulatory elements, some that are proximal, very close to TSS, some that are distant. Do they have specific epigenetic signature? Now for the simplest things was the study of their chromatin signature. You have, as I said, two types. How to find their chromatin signature. We did ChIP-seq with many different histone maps. Here is a heat map of, we focus only on the regulatory elements that are accessible after 1 hour of heat stress, not before, not after. And we check H3K4-Me3, H3K9AC, H3K18AC, and K27AC. So we saw almost no difference. Okay? You see a pattern, and this is how it fits with the proximal. But when we look for the same histone marks to the distal regulatory element, we found no K4-Me3, an increase of K9AC, an increase of K18AC, and almost no changes on K27AC. What does it mean? It means that you have specific histone modification associated to distal regulatory element. This is something that was not known at all. After that, what is the major or the main, let's say, role of regulatory element? Is to regulate the target gene. If the regulatory element is at more than 1 kb, 2 kb, sometimes 10 kb far, then you need to make a loop. But what is the role of this enhancer? Is to help recruiting the pole 2, or stabilizing the pole 2, which means that you need to have RNA pole 2 on the promoter, but you need to see it also on the enhancers. We did a chip to confirm if this is true or not, because this was not known in implant. And we saw that in the regulatory element, in the distal regulatory element that are active only at 1 hour, you get also the pole 2 on it, or at least an increase of pole 2. You have a little bit of it, but you get a strong increase of pole 2 at 1 hour also of heat stress, which means that we have found enhancers, found the chromatin signature. And here is an example of the type of data we generate. Here you have one gene in the minus strand. Here you have one gene in the plus strand, which means that the promoter here is here, the promoter here is here. You have all the data we generate. RNA-seq in control condition, almost no expression, a little bit here. Sorry, ATAC-seq in control condition, almost no accessibility except a little bit here. RNA expression, here a little bit expressed, here not expressed at all. After 1 hour of heat stress, the accessibility changed massively here, changed a little bit here, and you have a regulatory element in between them. The RNA-seq, you see the difference for the blue. Here and here, there is nothing here. Massive expression. After 6 hours, the accessibility decreased, and here is the pole 2, the different histone maps. But one of the key questions, you have here one regulatory element. It's in the center in terms of distance. And then the question, is it interacting with this gene or with this one? And the question is how to answer, because what I showed you with the high C, we didn't reach the resolution that allows us to answer this question. Then we set up in the lab an alternative of the high C, which is the capture high C. You do, in the beginning, the classical protocol of high C is just at the end. Instead of analyzing everything whole genome, we focus on the promoter, the tomato promoter. Then we capture all the promoter in tomato. We selected 1.5 kb distance of all the promoter. We designed 230,000 of probes to catch all the promoter. And what we found? We found basically that this regulatory element is able to interact with this gene and with this one, which means that it's not one enhancer for one gene. It can be one enhancer for two genes. And we saw, in fact, something even more complex that we try to make it as simple as possible. We found two types of interaction. Here you have one gene. This gene is not expressed, not accessible. Here you have a regulatory element that is far. There is no connection, physical connection. After 1 hour of heat stress, this gene gets up, the accessibility gets up, and you get an interaction with this regulatory element. But not only with this one, you get also interaction with some regulatory elements that are very close. We call this promoter hub model, which means that you have several regulatory elements that interact with one gene. We figure out also that you can have another model where you have several genes that will interact together and that we share few regulatory elements that we call the enhancer hub model. Okay, this is nice to describe something, but what I show you until now is pure correlation. And we wanted to go through functional analysis to try to understand what could be the protein that will promote the interaction between enhancer and promoters. Of course, you do the literature. You have a question, you check if others have answered this question before you. We did the literature, and some people propose that transcription factor could play a role in animals in this process. And then we say, okay, why not in tomato in our context? And the question is then, which transcription factor could be important? What we did, we took all the distal regulatory elements, we looked for motifs, and we were interested about the one that will be active only after 1 hour of heat stress, like the primary response. And of course, the motif we found is the HSF. As soon as we saw this, we made a bibliography. And we went to Akis, because he is the one who published all of the work with what is his name? The German guy with who you were working before. I don't want to say Klaus Wittgenstein, exactly. And then we went to their publication, and we saw that HSF1 in tomato is the master regulator. Then we went to him, asking him for the mutants. But before to show you the work with the mutant, what we did in the lab, we cloned HSF1A. No, he sent us the clone. We didn't clone it. He sent us the clone, and we look for the binding site and how to look for the binding site. We didn't want to transform tomato because it takes 1 year. And at that time, we didn't know how to transform tomato, to say the truth. And then we did what we call a DAP-seq. DAP-seq is basically you produce in vitro your protein of interest, and you mix it with DNA, and after that, you make a pull-down assay, and you get the DNA that can interact with your transcription factor, you sequence. We did this, and we found, of course, all the putative targets. Okay? It's different from the gene. It's the putative targets with the right motif, of course. And then we said, okay, before to move further, we have to have some good indication that HSF1A could be a good candidate gene. And what was the hypothesis of the assumption we made? We say, okay, we have distal regulatory element and gene here. We say, okay, if HSF1A is important, every interaction between distal regulatory element and a target gene of HSF1A must be much stronger after heat stress than in control condition. How to control this and to know this? We use the capture high C data, and we made this plot that we call aggregate plot. What is an aggregate plot? Is the average of high C matches for a set of regions. Basically what it says. We took, here is all the target gene of HSF1A. Here is all the distal regulatory element. If they interact, you see something red in the middle. If they do not interact, you don't see the red. This is in control condition. This is after 1 hour of heat stress. When we saw this, we said, wow, then it must be true. But since we are scientists, we have always doubt, and I was thinking that this could be due to artifact or I don't know. Then we use another technique that is a little bit different. We call it a high chip. What is a high chip? It's like the high C, but instead of looking to all interaction, we focus on the interaction associated with the RNA equal 2. Why? Because I showed you that the RNA equal 2 is sitting on enhancers. And then we wanted to see from these data if we can still get increased interaction between the regulatory element and target gene of HSF1A after 1 hour of heat stress. And this was the case. When we saw this, we said, okay, we should go for genetic manipulation and using like the tools that Akis sent us. What was the tools? He sent us RNA line because, as I told you, everything until here is pure correlation. And with just correlation, you cannot conclude really. You can make hypotheses, but you cannot conclude. And we wanted to really say if the transcription factor is important. Then here you have wild-type plant, 2 independent HSFA1A RNAi line. First, what we did in the lab, we confirmed, of course, that HSFA1A is downregulated. Second, we wanted to see if those clones has a phenotype. After 6 hours of heat stress, they start, to feel really bad. And of course, you cannot use this type of material to do like fancy molecular biology techniques. Then we did everything after 1 hour of heat stress, because at 1 hour of heat stress, we don't see anything change. We don't see like phenotypically changes. Then after 1 hour of heat stress, we say, okay, we take the wild type, we do RNA-seq in wild type, RNA-seq for the 2 independent HSFA1A lines that Akis sent us, and we found many genes that are downregulated in the mutant in the RNAi line compared to wild type, and those genes are associated to heat stress. Then this shows us that the material that Akis gave us is just perfect to do what we wanted. Of course, we have to control everything. I trust him, but as a scientist, we have to do it. And then we ask the question, to what extent the dynamic of HSFA1A regulatory element loop formation is the consequence rather than the cause of HSFA1 activity? And how we did this? I give you an example here of a gene that is well known to be very important for heat stress response. This is HSFA2. This gene, in controlled condition, is not expressed. The accessibility very low, except here. And here you are, let's say, 20 kb far from the promoter. If you look to the RNA pol 2 signal, which is in purple, you see almost nothing. Of course, it's not expressed. If you look to the capture high C data, you see some interaction, of course, but nothing that looks having any sense for us right now. And the high chip with pol 2, there was nothing, because no pol 2, no possibility to capture interaction associated with pol 2. After 1 hour, this gene is a target of HSFA1A, and this gene is upregulated. You see here, nothing here. The accessibility increased. The RNA pol 2 massively there. And then, if you look to the interaction, you see interaction with something here. If you look here, you have only 1 peak. Here you get 2. If you look to the pol 2, you get 2 peaks of pol 2. Those things are 2 regulatory elements. And this was confirmed with the high chip with the antibody against pol 2. Now, what we did after showing this? First, we cloned this. We cloned this, put it with the Mini35S promoter to check if this can drive transcription. Because if it is a enhancer, you put it in a Mini35S, you must see something. And then this is what we saw. And then after that, we saw, okay, let's use the line that Akis sent us. If you have this interaction, you must be able to see it in wild type, of course, and you must see this interaction in wild type after 1 hour, and this must be much longer than in controlled condition. Here, what you see is controlled condition. Here is after 1 hour in wild type. And here was just a negative control. And in the mutant, you don't see any more this interaction, which means that if you miss HSFA1A, you cannot create this kind of interaction between enhancer and promoter, which means that this transcription factor is key to allow interaction between distant elements. Of course, after that, we came with a model. We published this work last year, where in controlled condition, you have small nuclei. The chromatin is not densive for transcription. The regulatory elements are completely closed. After 1 hour of heat stress, you get the regulatory element accessible. The transcription factor goes and helps to form this type of loop and regulate gene expression. I will finish just by showing you the perspective of this work, because one of the key questions, I show you here, in controlled condition, everything is closed. After 1 hour of heat stress, then it suddenly becomes open. One of the key questions, how this can be? And one of the things that we know from the animal field is that some transcription factor could have the capacity to bind to chromatin, to bind to DNA, even if the DNA is associated with the nucleus. We call them pioneer transcription factor. And we ask the question, is HSFA1A could have this capacity of being a pioneer one? How we went to this idea? Because we did the ATAC-seq in the line that Akis sent us, like the RNAi line, and compared it to wild type. And we focus on the HSFA1A target. We found some targets for which you have no differences in wild type after 1 hour or in the mutant after 1 hour. But we found also several targets where if you miss HSFA1A, those regulatory elements cannot be open, cannot be active anymore. I show you just a screenshot here to give you an idea of what type of data we are dealing with. All the arrows are the genes with the orientation of the genes. Here, the pink is the binding site of HSF1A, no accessibility in control, accessible after 1 hour of heat stress in control. But in the mutant, here it's the orange one, there is nothing. And the gene is down because you need HSF1A to be present to open this. And this leads us to ask the question about HSF1A. Is that if we could have this activity of a biological factor, and we are working on this right now. Now I finished because I'm over the time, I think. I would like to thank all of the people in my lab, the ChromeD team, the PhD student who did this now, she's in Austria doing a fancy postdoc. I need to thank my collaborator, Jose Gutierrez Marcos, who is here. Akis also, I will never try to pronounce your name, no? Too complex for me. And the people who gave us money to allow us to do this work. Thank you very much.
I would like to thank Musa for this fantastic presentation. We have time for one question, and then Musa will be available for more questions. Irene?
I raised this, but I have 3 questions, but I want
Only one.
that is more specific. And so do you think it's only HSF1 age? Have you ever considered doing the opposite vaccine to try to
Yes. We tried this with Jose. We tried what we call the reverse cheat to find the protein that binds. We didn't succeed right now, but we didn't give up. We hope we will be able. And of course, it's not only HSF1A. We think that HSF1A is a master regulator, but of course there are many other transcription factors that we really think could be. Yes. You think you'll be right, and we try to study this.
Okay. Thank you, Musa. So let's go start with questions to Musa.
Thank you. Thank you very much, Musa. And our next speaker is Mr. Sotirios Stefanakis. I did it, yeah.
He writes in the right way in the papers.
Yeah. Before yours.
Okay. Thank you very much, the organizers, for giving me the opportunity to present the work in my lab. I don't thank them for putting me up with this amazing talk, so I'll bring you back to earth with this talk. And I will change a little bit. I will stay on the same plant and on the same transcription factor family, but a little bit different. So I'll talk about class B HSS, and this is something that we don't talk. So my lab is located in Frankfurt. These are, I don't like this picture because I don't know, I look like a king, but whatever. The photographer told us to do like this, but I have this fantastic team from PhD student, master's student, PhDs, and technician. And what we are trying to address is how plants respond to high temperatures, what are the mechanisms that they use in order to survive stress, and what makes a plant more tolerant than another plant that is more sensitive. And we work on different levels, from the cell level to the organismal level, from seedlings to mature plants, and even more agronomically important plant parts like reproductive tissues. And we are focusing on addressing 2 questions related: heat response, transcriptional regulation, so a lot by Musa, but the last years we are also focusing a lot about splicing. I love the subject, but I will not talk about it today. I will talk only about transcription. Now, already Musa made a very nice introduction. He told me only about one HSF, but the reality is that it's much, much more complex. So HSFs are large families. In plants, most of the plants, they have more than 20 members. In tomatoes, there are 24 members, so Musa can feel very lucky that in tomato, only one is a master regulator, that's all the important work that he presented. But there are another 23. And HSFs can be classified in 3 subclasses. They're called A, B, and C. They're all having a DNA binding domain to bind with these heat stress elements that Musa presented. They have oligomerization domains. I will come a little bit later to this. And one thing that differentiates between A and B is that class A HSFs, like HSF A1A, they have activator motifs, so they are important activators. And class B HSS, they don't have this activator domain, but they have a repressor domain. Remember, transcription is important for transcription activation, but also repression. Class C HSF, it's enigmatic. We don't know because they don't have activation domain, they don't have a repression domain. And I'm happy because the last year, the HSF tomato family became much bigger. So at least through Epicatch, I'm able to talk to several people. Musa, you already showed, he's doing all the fancy work that I'm not able to do in the lab, but answer the big questions, what happens on the global level. I'm very happy because Michal, who unfortunately is not here today, she asked me, what about class CHSFs? So we are working on this, and I have lovely Irene sitting here in the first row, who asked me, what about light and heat stress, because they coincide as you go out, you will realize today. So we have this team working all together on plant HSFs. So as Musa showed before, with activators, the story we have elaborated a lot in my lab. Already, Musa showed that class HSF-A1A is the master regulator. So if you put plants under heat stress, A1A is activated, it binds to promoters of many genes, including heat shock proteins that are essential factors for survival under heat stress, but also they activate other HSFs, including HSF-A2 and A7. And we have shown in several publications that plants are able to produce A2 and A7, and along with A1, if there is a recurring stress, they can form hetero-oligomeric complex with very high transcriptional activity, and under conditions that are very strong for transcription, and they are repressing actual transcription, the presence of these activator complexes allows response, and plants can survive, so they are acclimated. And these are important also for memory. And now I would like to switch. And we are talking always about class A HSS because they are the activators, but you should always remember that the repression, the attenuation of the stress response is very important. If you make a heat stress for 1 hour and you follow the HSP mRNA levels as markers for HSF activity, you will realize that they reduce very fast during heat stress, and within 1 hour they peak, the expression peaks. If you put back the plants to recovery, 25 degrees for tomato, you will see a very fast attenuation because these mRNAs are very sensitive, they are degraded very fast. But in contrast to what many people expect, which are not from the field of heat stress, if you extend the response, the expression will not continue, the mRNA synthesis will not continue to follow the temperature. You will see that the stress will attenuate regarding the production of HSPs. And this is because the mRNAs are there, they can be used for the synthesis of proteins. And the plants, for the plants, it's very important to keep a balance. Something is here that is missing, it doesn't matter. It's a balance between these two processes. From the one hand, plants, they want to induce the response which will help them to protect themselves and to survive. But on the other side, these protections should not come into what's the English word? expense to development and growth. Always the stress will stop, and then the plants they have to recover. So attenuation is very important. And I have an example here from 2 tomato plants. This is a wild type plant under control conditions. There is no heat stress response. This plant, if it's exposed to heat stress, it will be sensitive, but it looks healthy under control conditions. This plant here is a transgenic plant. Heat stress response is constitutively active. These plants can survive very strong heat stress treatments, but the plants does not look happy at all. So, thermotolerance and overall health balance is very important. So it's not only about activation, it's about attenuation of stress. Who does this job? For the attenuation, class B HSFs. They have the repressor domain. You see that if we take one of these class B HSFs, which we call B1, A1 acts as an activator, this is a gas reporter assay, B1 acts as a repressor. If we combine both A1A and B1, we co-express them, we see that B1 competes the activity of B1 and repress the master domain. This happens for A2 and A7. I told you before, these are important for the accumulation, important co-activators of A1 key. So it's not a surprise that if we have transgenic plants that we suppress the expression of the repressor, expression of target genes of HSFs, including other HSFs or HSPs, is higher under controlled and heat stress conditions, and these plants are more thermotolerant. Now, and I will show you only unpublished results from now on and what our ideas are for these models. How this could work? We know already that the repressor domain is a tetrapeptide, so they are LFGP peptide, and if we delete it, B1 loses the repressor. Why this is important? Because LFGP is a very conserved repressor domain in many transcription factors, and it's known to interact with topless genes, very important for repressors. So what we think is that B1 requires topless requires a histone deacetyltransferase, and probably this leads to the repressor, transcriptional activity. Now, at the moment, what we have done is that we have cloned all topless genes in tomato, we co-expressed them with B1. So you see B1 has a repressor activity, this topless is also a repressor activity. If you co-express them, the repressor activity is lower. So they both repress this gas reporter assay based on HSP. What happens if we knock down topless 6? We lose the repressor effect of B1. So we think that B1 interacts with topless 6 and probably this to the histone deacetylase, removes the acetyl group from the H3K9, for example, that was associated before, and this results in repression transcription. Okay, so this was very expected because we have a repression domain, there are repressors. But as if you remember the time, I talked about a molecular switch. Why a molecular switch? Because if we take other promoters, like this from the small HSP17, if we express A1, it acts as an activator, B1 as a repressor alone, but if we combine them now, the activity is much stronger than even A1 alone. And the coactivity is even much stronger from the coactivator superactivator complex of A1 and A2. So they are not only co-repressors, are also coactivators. And now, this was confusing for us. Why? Because if you see here, one structural difference between class A and class B HSFs is in the oligomerization domain. Class B HSFs, they have a very compact oligomerization domain, while class A HSF, they have an extension. In the middle, they have a linker of 21 amino acids. What is this oligomerization domain? It's a classical coil zipper motif with every 7 amino acid leucine. And this compact, this extension here, does not allow a perfect match to make zipper oligomerization domains. What does this mean? That these two, they cannot physically interact. A and B cannot interact. So how does this happen? I'll show you in the next slide. What is the concept of a coactivator? If we overexpress B1 as a coactivator, it will lead to the overaccumulation of HSPs and to thermotolerant plants. So to repress B1, thermotolerant. You overexpress B1, again thermotolerant. And we did also transcriptome analysis, and we found many genes that are regulated by B1. We did under controlled conditions, after 1 hour of heat stress and after 1 hour of recovery. And we saw that the repressor and coactivator activity is based on different conditions. So you can find some genes that are affected only due to heat stress from these activities or during the recovery under controlled conditions. They are gene specific and very important. They affect heat stress induced genes, but also constitutively expressed genes. So it seems that these class B HSFs, they have a broader role than the classical class A activator HSFs. So we did also transcriptome, in addition to the transcriptome, we did proteome, metabolome, and hormone analysis to understand a little bit the physiological role of this HSF. And what we found is that it doesn't only affect protein homeostasis through HSPs, it regulates many histone regulating genes, many photosynthesis-related genes, drought stress. So maybe it's a hub for different abiotic stresses, but also biotic because it regulates many pathogenesis-related genes. Through the metabolome, also we saw that phenylpropanoid pathway is affected and salicylic acid and jasmonic acid. So we think that we have an HSF that it's in the hub of different stresses that in nature they co-occur heat stress and drought stress, heat stress and biotic stresses. Okay, so for the co-activator, because it's very enigmatic what happens, there is a screen with different mutations, and we found out that in the C-terminus of the protein there is this GRGK motif that is important for the co-activator. As long as this is mutated or is deleted, then the co-activator activity is lost. So they cannot interact, but still they can make a complex. How they do it? There must be probably a third protein that acts like a bridge. And this protein was found to be a histone acetyltransferase from the CDP P300 subfamily of histone acetyltransferase. This is called HAP1, and we have confirmed by two ways that HAP1 is important. So all these three make a ternary complex. If we co-express A1A with B1, we get the co-activator activity that I showed you here. But if we add a CRISPR, a plasmid that has a CRISPR gRNA, this co-activity is lost. And we did also transgenic lines, mutant lines, CRISPR lines, that HAP1 is not expressed anymore. And now if we show a gene that it's heat stress induced, 2 minutes, yes, HSFA2 is not induced anymore. Okay, so HAP1 is a histone acetyltransferase. We know that it interacts with A1A and B1, it makes a ternary complex, and is important for heat stress response. And to go a little bit faster, so this is our theory. Of course, it's already known. Also, most have talked about that histone, H3K9AC, for example, is a positive marker for expression of genes under heat stress. We can confirm this for several genes. In the overexpression line, we saw that there is an enrichment of H3K9AC in several heat stress-induced genes. And also, recently we did a proteome analysis of the HAP1 mutants, and we found that there is lower levels of H3K9AC, also 18AC, and H4AC. So it is an histone acetyltransferase. So we think that this ternary complex brings the histone acetyltransferase and the promoters of heat stress-induced genes. These are acetylated and they use even more the expression of these genes. Okay, so I hope I convinced you that there is a molecular switch. There is HSFB1, HSFB1 acting as co-repressor or co-activator. Now, HAP1, and I will go in 1 minute with this, is very important not only for heat stress response, but as you expect, also it has developmental role. So the HAP1 mutants, they have 4 cotyledons compared to the wild type, it's only 2, they are shorter, they have these fasciated stems, also the morphology of the leaves is altered, they have too many leaflets and larger leaves. The time to ripen from aphesis to ripen is delayed in the HAP1 mutants, but to our surprise, the time to overripe is much, much shorter. So we think, and again something is missing from here, is that this HAP1 is the hub between what I told you before, protection, survival, recovery, and development and growth. And I will give you this example and I'm done, because till now I did not show you that any class B HSF has any heat stress and developmental growth, but it's important to connect these two. So tomato plants, the wild tomato species, they have an extended stigma. This is very common because it favors cross-pollination. During tomato domestication, there was a selection which is based on 2 genes, but the stigma became inserted because the style became shorter, because this gives higher production and it avoids cross-pollination that breeders they don't want and farmers they don't want. But still, some modern cultivars of tomato under heat stress, they have an elongated style. Okay, and this results in reduction of production. One of these molecular switches, when we mutate it, it shows this accepted stigma under heat stress, which is here. And if we overexpress it, the stigma becomes shorter, it's even more inserted, and this results in higher food production because of higher fertilization success. So by this, I hope I convince you that class A HSS with the master regulator are important. They are probably the pioneers for stress response and thermal tolerance, but if you want to understand how plants respond to heat stress, we should also have a look at class B HSS. And I would like, first of all, to thank all the beautiful people that are working with me in Frankfurt, the collaborators, Musa, Irini, Michal, all the people here, local organizers, and you for listening.
So we have time for one question.
Okay, just a very general question. I just wondering if you have looked at this answer at the organ or tissue level. And can we say that what you show is general for all the type of organs in the response of this stress? And coupled to that, because you just mentioned two hormones, but problem of maturation, is there anything to see with ethylene?
Okay, thank you for the very nice question. So, is this general? No, because when we take the lines that Musa showed, the RNAi, the master regulator, we do not show the last phenotype. So it seems that this HSF that has this phenotype is independent of the master regulator. Some things like the production of HSPs are dependent on one gene, it's the pioneer, but other HSFs, that's why we have this shoot of 24 HSFs are needed to induce different responses. If we have seen ethylene, ethylene is not involved in thermotolerance. It's a very important hormone for stress response. We did not check ethylene, but for sure there is a role. So we have some mutants with ethylene receptors, and we are checking this now. But definitely it should be around.
Okay, thank you, Arthur, for the beautiful presentation. And now next speaker is Federico Martinelli.
Which one is it? Okay.
Okay, so today I'm gonna talk about the work that I actually did in a short-term scientific mission of Epicatch. I did not do the work. As you see, it's a very long work. It's more than 10 years of work. I just went there to my collaborators, my mentor in UC Davis for a short time, and I'm just analyzing the data that he obtained. Okay, so yes, okay.
It's working.
The work, as you see, is dealing with use of RNA interference in order to suppress crown gall disease in worms. That is caused by Agrobacterium pneumophages. As everybody knows, is the model of study in plant transformation, because for more than 40 years it was found that this bacteria was able to transfer genetic material between different organisms in different kingdoms. And also, Agrobacterium is a model of study in investigating mechanism dealing with horizontal gene transfer, but also type 4 bacterial secretory systems. But the use of Agrobacterium pneumophages in plant transformation basically overshadowed his serious role in making a different disease in different crops.
Agrobacterium, as you guys know, is a bacteria that is present in any kind of soil. Most of it is present in the kind of organic matter, and as I said, is agent of many diseases in different crops. For example, species of agrobacteria are causing hairy roots disease, but there is also other species like Agrobacterium vitis, the cause of crown gall in grapes. Basically, this disease caused by Agrobacterium, most of them they are not fatal, but they can be dangerous when they infect young materials, and they are associated with a reduction of crop yield and vigor in, as I said, different horticultural crops like apple, grape, and cherry, in addition to walnut. What are the mechanisms of this disease? There is still a debate on the scientific community on what is the cause of this kind of disease, and most of them think that the cause is decreased water and nutrient supply to the infected plant, because water and nutrients goes to the unproductive gall sink. And also, infected plants are also very susceptible to secondary infections and also other abiotic stress, and also there is a negative effect on graft take. Agrobacterium tumefaciens as an agent of disease has been addressed by different strategies in order to try to control this kind of disease. And there were different strategies trying to address the different triangles of the disease triangle, like the host. And so in literature there are different works dealing with the development of resistant germplasm, trying to silence oncogene genes. And there are also literature dealing with trying to work on the environment, reducing bound and make phytosanitation, and also there are strategies directed to the pathogen. Our strategies was directed to the host using a transgrafting solution. Transgrafting, what is transgrafting? It's just the combination of known transgenic scions in transgenic rootstock or vice versa. And transgrafting, of course, is already have been used in order to control other environmental stresses. What are the benefits? Basically, when you use a transgenic rootstock, you prevent the transgenic flow. As I said, was using transgrafting. So scion were transgrafted in transgenic rootstock, expressing double-strand RNA directed to two pathogen genes, tryptophan monoxygenase and isopentenyl transferase, with the aim of control the disease, determine if the double-strand RNA is transiting to the scion, and also understand the metabolome and proteome effect on the scion, and investigate if there were other side effects on disease-related traits. A few words about the experimental design. As I said, the construct expressed a double-strand RNA directed to these two genes, and the plants were transformed and transferred in the field, and several lines were screened. And then two lines were selected because they didn't show any crown gall in any plants. And so we investigated more on the detail, the effect on these two lines. So there were two kinds of experiments. One experiment, we compare one transgenic line, one transgrafted plant against a control. tree, just a grafted wild type tree, and we analyze small RNA, proteome, metabolome, and then we have a second experiment where we use the other transgrafted plant against the control, and we try to analyze more or less the same thing. As you see, we basically analyze the bark tissue, the tissue that is above the graft union, that is with the scion, and below the graft union, that is the sock. And in the second experiment, as you see, we analyze also the effect on the nut. Here is just a picture, just to make you understand what we did. The focus was on the bark tissue above and below the graft union. The results. So basically, the small RNA sequencing did not show any double-strand RNA in the scion tissue of the transgrafted traits. As you see there were no reads mapping to the scion tissues. That means that this double-strand RNA doesn't move in the scion. We map the RNA read against the pathogen gene, agrobacterium genome, and as you see, there were only reads against rootstock and graft union, no reads in the other tissue on the scion and the nut in both experiments. As you see here, also we checked if there were reads against other transgenic components, the resistant gene, canamycin, and the gas promoter, and again, no reads in the scion, only in the rootstock and in the union. Relating to the other effect on disease-related traits, no significant effects. Comparing control and transgenic plants, no significant changes in any other disease involved. So basically, no other effect in addition to the crown gall disease. What about the proteome? The proteome was analyzed with iTrack system. We analyzed more than 2,500 proteins, and when we compare scion, the rootstock, and the graft union, control and transgrafted plants, very few proteins were only identified for the transgrafted or the grafted. So basically, no big changes on the proteome. And one important thing was the fact that we observe only the transgenic protein, the gas, and the canamycin resistant protein only, again, rootstock and graft union, no in the scion. Relating to the metabolome, here is the PCA plot of the metabolome. Basically, as you see, there were not too many changes, and so we were not able to separate well transgrafted and grafted. That means that the metabolome does not change too much in relation to the transgrafting. But it's possible only to separate the samples based on the different type of barks. Okay, so this means that transgrafting probably doesn't have not too much effect on the metabolome of the bark. And again, the different pairwise comparison were did, and we took a look on the few metabolites that were changing in the scion tissues of wild type and transgrafted plants. As you see, anyway, this pairwise comparison, scion, is the one that has less number of metabolites. That means, again, that scion is not too much affected. And this also in the other experiment with the other transgenic lines. Again, this comparison has a very low number of changing metabolites. Anyway, it was interesting to take a look on the few metabolites that were changing. in the first transgenic line, they were basically related with secondary metabolites like kainate, cyclate, and phenylalanine that were induced by the transcripting, and everybody knows they are related with oxidative stress. So we should do maybe further investigation, see if these transcripted plants are more resistant to oxidative stress. Another interesting change was related with serotonin. As you know, serotonin has a very important role, healthy perspective. Walnut is rich on serotonin, but serotonin is also linked with biotic and abiotic stress responses in plants. We observe a change of serotonin in the bark tissue, but not change in the walnut nut. We did also the gene set enrichment analysis in order to see the metabolic pathways affected by transcripting, and TCA cycle was the category that was more repressed in the cyan bark, and aspartate family was the one that more was repressed in the graft union. Then we try to integrate proteome and metabolome. Here in the picture is a pathway that we focus our attention, carbon-nitrogen balance. As you see, there were several genes that were down and up regulated, and we paid attention on some metabolites like glucose, maltose, and glutamate that were affected by transgrafting. The second experiment, basically, as I said, very few metabolites were changing. They were related with flavonoids, and they were upregulated in transgrafted trees, and also phenylalanine. This one was found also in the previous transgrafted line. And finally, also in the nut, we analyzed the metabolome of nut tissues, and we basically compare the pellicle and the kernel. And as you see, there were changing metabolites when you do the pairwise comparison between tissues, but when you compare same tissues of transgrafted and grafted trees, very few metabolites were changing. Again, this is a demonstration that basically transgrafting helps to suppress the disease, but doesn't affect too much the quality of the sign. And so, in conclusion, this work shows that this strategy was helpful in order to suppress the tumor. So RNA interference can be a good strategy for transgrafting in order to control disease in walnut. And also this study shows that there were no transgenic double-strand RNA movement in the cyan. In the literature, there are other data that shows the opposite. And also the transgenic proteins was not moving in the cyan. It's another important finding, and the few metabolites were affected. And then so this study can confirm that basically this strategy can solve some food and public opinion on concern of transgenics. And as I said, the work was done at UC Davis. Here is Abhay Landkar, my mentor in UC Davis, and this is his group.
Thank you, Federico, for this very interesting talk. There's time for questions. Here. Any question? So can you say a little bit about the timeline of this short-term scientific mission?
So basically, so this is something that really I want to say, because I think a short-term scientific mission are really important. I just came there for 2 weeks. my aim was trying to publish this data. It was not possible. Because
but for the analysis, the short-term.
the analysis was great. So in these 2 weeks, I'm focusing on analyzing, writing the work, and we try. So I have a paper, a draft of the paper that is sitting there. So I hope.
But I meant about the time. How old were the plants when this experiment started? I mean, I understand the analysis. So short-term scientific distribution can be for the analysis results, obviously.
It took more than 10 years.
Yeah.
Basically, as you know, walnut is really it's not very hard to transform, but to put in the field and analyze the big, huge work that basically Abaya has did for more than 10 years.
Yeah.
I don't know if I understood correctly. Correct me if I'm wrong, but I mean, from your data suggests that transgrafting may work, right?
Yeah.
But what you are telling us is that you haven't been able to see what is the possible mechanism that underlines these or underpins this phenotype, right?
Yes.
Could it be the timing of when these samples are being taken? Like, you know, if you're taking the samples for the small analysis and it's late in development, maybe everything has happened, right? Could it be, or?
So, okay, so the effect was clear. The crown goal was not occurring in the transgenic rootstock. Okay, so because the goal is on the rootstock. Okay, and we analyze basically what we wanted is to see the effect on the bark tissue of these transgenic plants that didn't show the crown goal, because the question was what happened to the sign. Okay, because the goal is occurring on the transgenic roots, on the roots. Okay, so the goal is on the, let's say, the roots. Okay, not on the sign. The scientific question was what are the changes on the science related to the transgenic components that is expressed on the rootstock. Okay, and basically, as you see, not many changes on the proteome and on the metabolome, very few metabolites. So we think that this can be a very good solution to control this disease because it's a disease of the roots. Okay, and the product, the transgenic product, doesn't go on the sign, so there is no concern about any movement. That was basically the question. That's why we analyzed the bark tissues on the graft union in order to see if the transgenic components was going to the sign. The mechanism of the suppression was basically a mean or aim. Okay, so what we wanted to address is what is the effect on the sign, and the data shows that there are not so many changes.
And maybe can you say for people that they don't know how they can apply for the STS7?
Yeah, we should apply. So we have still 3 months left, as you know, and we have a lot of budget. So please try to apply, because so basically what you have to do is contact George. I'm not seeing George.
He will come tomorrow, I think.
He will come tomorrow, yes. So George is the manager of the short-term scientific mission. So you still have time to apply online in Costa for a mission, whatever you want to do it. Basically, they are up to 1 month, so you can spend up to 1 month, and you can visit any places. Obviously, dealing with the plant epigenetics.
Yeah. All right. Thank you, Federico. So now we will have a coffee break, half an hour, and we will start and be back at 16:30.
Tomorrow's here.
So 16:30 sharp, all right? Just follow the signs. Thank you. That was a lot. Pleasure.
Possible to switch off the lights close to the screen?
Yes, yes.
Otherwise, have some microscope
Okay.
Come vedete dopo.
Okay, okay, I will ask my professor. But it sounds possible. Okay. Have something
Yes, I am.
So this one is yours, yeah?
You will take it. Yeah, I just want to close the present.
Yeah.
Yes, it's here. I just wanted to provide you this in case
Here you go.
we need to charge it.
Ah, all right.
Just remember to add Alexandre on the
Yeah,
program.
yeah, it's already.
And just
we working
tell
on
him
it.
what will be the time, because finally it's out of the time. For the battery.
Sure.
Just also remember, if you edit, that Michal is not there, so tomorrow afternoon there will be a 20 minutes left. Maybe you can go to the talk without changing a lot of things.
I believe.
Yeah, now it will be charged.
Well,
This is the yes, and we can yes, you turn it on. You wait a second to connect, and then it will.
It's okay. Somebody will be here.
Besi, a ti si tamo ostavila bila?
I think it's safe here.
Koga slučaj? Postići kako se razabili. Joj, kako sada zaključam?
Yeah, okay.
Okay.
You will be here? Are you going to stay here?
Yeah, no, I'm going to get the mobile and then I can go outside.
No, it's fine. It's fine. Just, yeah.
You wanted to close, no? To lock the door.
Yeah, but I don't have the key. So all the time someone's here because of, yeah. That's there's no one staying.
I wanted to check my slides because I use a Mac and I always, yeah. Just to say, that, I mean, the movies are not working, but they're not important.
It's possible to switch off some lights.
Yeah, I agree. For all your images. Huh?
Yeah, I have all the lights going.
Mm.
Maybe tonight, something like that will be okay.
Yeah, that one needs to be off.
Yeah. If they have high a lot of features with the microscopes, I need a headphone. That's really fun.
This talk is also online. People are participating online.
I don't know, actually. It wasn't supposed to be a hybrid.
No, because I couldn't see anyone like joining, but they are using Zoom, no? So
Because if are people joining, then we should check if there are questions or the
Yeah.
something, no?
Yeah.
I don't know. Everything's impossible to read for me, so.
Sarah, did it take forever for you? Did it take a long time to from slide to slide? Or is it just my images?
I don't know. I haven't checked mine, so I want also to check mine.
Right. Yes.
Have to try to put them in full screen and see how it works.
Because it takes a long time to change from slide to slide.
Okay.
You have many slides open or many talks open?
It's only mine, I think.
Oh,
yeah.
Yeah, because I'm the first one after the coffee break, so I think that my presentation will be ready.
Anyway, we'll figure it out.
But it's true that, I mean, my presentation is quite big because I have all the images. So.
So I will minimize mine and I want to check it. And I don't know how the
Okay. Seems okay.
yours are changing.
Oops. Okay, the animation color don't work here.
Yeah.
Small screen for the big room. Um, So I have an animation here. No animation. I don't understand why
things move in your presentation.
But I didn't want to have animations. I'm not completely sure why I have them. So everything should be from the very beginning, but I don't have. You see, this transition. I don't understand. So if I say, so if I go to animation, this doesn't appear like it's animated. You see?
Yes, but when you change slides, it comes as
It
animation.
comes like an animation.
Oh, I should
yeah.
check that too. That would be
And
interesting.
it's also changing me things like this. I don't know. I mean, that's a small thing, you know, but it doesn't look like my computer, you know. But I cannot. I don't know. If I have animation, it should appear like here, no? Like you have something,
exactly.
but
One, two, whatever.
yeah, 1, 2, 3, but nothing appears. So I really don't understand. And it says no animation.
So animation on the book?
No,
There shouldn't be one, but
it shouldn't be. I don't have any animations, but it looks like, you see, it's very strange. So everything should be there from the very beginning.
Oh, I should check that. You're right. Yeah.
And I don't know.
I think it's just the picture is loading slowly.
That's the problem. Yeah.
There's no animation.
No, but no, it's very weird. So, okay, this is okay. This, but no, this is not okay. Really.
Can you connect your computer?
I'm going to ask for that. Yeah.
Yes, because that would be the easiest. I'll have a look for mine too.
I
have no idea. View. Or do you go to slideshow? Mine doesn't even change the slide. That's the problem. Look, I'm pressing it like a few times now. It will take a while. Yeah, there's a delay for loading. Oh. Hmm.
I asked the girl outside. Oh, I'm sure it's fine.
Yes, so what make one three.
No, it's not like the killer make one. It's not like 3, 5, 6. Yeah, I see.
That was very, very nice.
It's not working.
Yeah,
Yeah.